So I already can start talking because we don't really need slides to get the general concept across. So I'm here to talk about principles of brain organization, thinking about what to do once you have a nice parcellation, but you want to understand how the regions that are in certain places in the brain became on the location they are within the brain. So rather than understand the individual regions, we try to understand the interrelationships. And one analogy that I like to use is, you all know planet Earth, right? And the continental drift model. So if you look at Google Maps, you see the different continents placed across the brain. Sorry, this is like, you can also close your eyes, but you can see the slides later, but it's also to save time. So anyway, you have a map of the Earth, and you see all the different continents, but to understand how they came where they are, you have to understand the model of continental drift, because this also helps you understand how South America kind of fits into Africa, and also how the past of the Earth has shaped the current present. And knowing the model of continental drift, it's also relevant to know like fractures in the Earth, such as Iceland, and there's a lot of volcanoes, but also, what is it, the Andreas Fault, the one that is in the Pacific? And knowing these past and also these tensions between the different continents helps us to understand also what regions may be risky to some extent, because there can be volcanoes, but there can also be geysers. So it's also risky and fun, it's all together. So with this model, knowing this map, we can understand the risk, but at the same time thinking about the map, there's also other elements of the map, such as the weather that changes all the time. So context really matters a lot to interpret maps, and these are all can be explained from different principles. So bringing this back to the brain, and this is also why I think it's relevant to consider gradient and also contextual decoding if you look at individual regions. We have the brain that is made of a combination of genes and environment, and as Roberto also explained, mechanic factors, right? And what we do in the lab, and what we do also with a lot of our analysis, bridging different skills, is trying to unfold how a brain came as it is in a certain situation when it was scanned. So this is the tools that I'm trying to explain once the laptop is connected. Otherwise, it will be an interesting educational session. The other laptop also doesn't connect anymore. What is the next slide, actually? Okay, so we've passed the Earth, the general principle of continental drift, and a little bit how it could relate to the brain. So what is nice about gradients is that they represent many things at once, and they don't exclude the notions of areas at all, but they help us to understand why regions are in the reason they are, and they also link to patterns of development, but also patterns of function and of evolution and how it's changed. For example, differentiate sensory and transmodal areas. And also this links to myelination profiles, which is again also linked to neurodevelopment of how myelination changes from more sensory areas to more transmodal areas as a function of time. And there's different ways, like gradient is on the one hand also a method and a way of thinking about low-dimensional spaces. A very common method is some that you may know or not, but it's the gradient method of Daniel Margulis, although there's also other methods of the Beckman team in Nijmegen, and this can all be addressed also with a toolbox called BrainSpace. So I see stuff happening here, but at least this toolbox, you put in a connectome, and what you want to do is reduce the dimensions. So you use a PCA or a more nonlinear transformation to get the main sources of variance along the spatial axes. And this helps us to understand what patterns shape this organization in the functional connectome. So if you're already super curious, you can go to brainspace.read the docs, but we also have a tutorial. I see now people looking worried. Maybe we broke the room. That's we can also have a coffee break now, and then in 15, or is it? Yeah? Like, let's have a 10-minute coffee break. We fix the setup here, and then we continue. Yeah. Okay. So people are coming back, yeah? And I hope you had some coffee or tea. I think I'm here to say we have no disclosures. And also, thank you, Matt, for solving the problem. Plug unplug was the secret, I guess. So this is maybe the most educational element of the whole session. So yeah, I will brush a bit through the beginning because I kind of said it, but maybe it's nice for you to see the slides that belong to what I said, in case you have aphantasia, is something that we also discussed, that you don't see anything before your eyes. So why should we care about gradients or brain organization in particular? So as Brodmann said, cited here in Panja 2015, whatever the method, our archetypal approaches to understanding the cortex are based on distinguishing different laminar patterns in different cortical areas. Without question, this approach provides an invaluable framework for understanding cortical organization. However, up to this time, architectonics has not fully addressed an important issue raised by Brodmann in 1909, namely the search for morphological principles underlying the differential distributions in cells in the cortical laminae. So I guess, I don't know if you know the recent work also from Peterson and Gordon, where they were like a bit dissing gradients, which is totally fine, and I think there's also many interpretations where one could disagree with, but at the same time, I think there's also many other interpretations where you can also understand that it's important to understand the rules of the system. And there's many ways to understand this, and this is where I and Amin will talk about. So the human brain can be seen as a scaffold between genes and environment, producing functions. So on one hand, we have genes shaping the brain, starting both from an evolutionary perspective, but also an ontogenetic, so from birth on, or from conception onwards perspective, which is, what was it, 5 weeks, right? Yeah, 5 weeks. So I thought it was earlier, but good. But anyway, so from 5 weeks of time, the brain is shaped, and this helps us also to interact with the environment, both first in the womb of the mother, but then also once born outside of the world, and also the bodily environment. In turn, this interaction then also shapes the brain through shaping its function and then also creating certain organizational features, but also learning certain functions and behaviors. And in turn, this makes then also the context in which the expression of certain genes matter, both across time and space. So in the end, one can conceive this interrelationship like as an internal circle that we want to kind of unfold. And for me, understanding rules in chaos is a good access point. So this is the map analogy. So you see here a bit how it went. So first you have Google Maps, then you have the continental drift for the people that couldn't imagine what I just said. And here you have the Andreas Fault, saying that it's dangerous to be here, but also, well, kind of fun and interesting. And it's also generally also an area where the soil is very good to plant certain fruits. At the same time, a map and understanding how it relates to like how pleasant it is, cannot exist without understanding the dynamics or the environment / the weather. Because this can make a beach a very pleasant place where I trust my kids to be at the shore. First, it's a place where I don't want to buy a house and it may even be dangerous. So in analog to the brain, this is why we want to understand maps and also understand locations and the functional dynamics within it. So one example, for example, is a question that I asked now 4 years ago. What is the genetic basis of the spatial organization of macroscale cortical features? So for this, I used a gradient approach where I used the twins from the Human Connectome project to make a genetic correlation, a matrix from how genetically similar two given regions in the brain were. Then I created the principal component of this covariation to discover that regions with the same colors have a somewhat similar genetic profile, going from more posterior areas in blue towards the yellow areas here in the frontal cortex. And what you see, hey Daniel, you've missed the, no, never, it's a, I made a joke about him when I gave the same talk. He didn't like it. Or he did, I don't know. Anyway, so there is this spatial distribution going from more posterior or unimodal areas to frontal areas in the human. And what is interesting, if you look at genetics not only based on twin-based genetic similarity, but also from an evolutionary perspective. We also looked at open data from the prime data set, and in macaques we also performed a covariance analysis, helping to understand how similar regions were across the group of people, representing largely genetic features. And we did a gradient approach, and again we see this differentiation between more sensory/posterior areas towards more frontal areas in yellow. And importantly, with help of Ting Su's projection algorithm, we also projected the human pattern in macaque space to really do a spatial correlation between both patterns, and we confirmed that it was the same. So it's not just me giving it the same colors, but there is also a statistical relationship. And this pattern relates also to a differentiation of sensory motor to transmodal areas. A second pattern that we found differentiated more inferior regions from more superior regions, here in red and green. These regions were also differentiable in the macaque pattern brain, where you see a differentiation between temporal lobe and more visual but also superior areas. And what is interesting is that its pattern, when we made a model of the dual origin model based on the geodesic distance from archaeocortex and the paleocortex, which roughly corresponds to the hippocampus and the olfactory areas, we found that these spatial differentiation of genetic similarity related to this dual origin model that relates to waves of laminarization that are shaped in early ontogenetic phases, shaping up the brain from highly agranular areas towards corneal cortex. So in this study, I used a gradient approach to define two axes of genetic organization in the cortex, and as such, it may, in a map analogy, provide a compass to understand where we are in the brain and how it came to be that a certain region is where it is along these different quadrants of rules. And it's important to note that although I only highlight two rules here, one have to argue that there's many more principles that overlay together and thereby define specific features of a specific area. So that's how I see that it's both related. So yeah, gradients provide a coordinate system in the brain. For example, one relates to a functional hierarchy or at least a functional differentiation. If you look at meta-analytical maps of task activations, you can see that there's a differentiation between the regions that are meta-analytical, more active during motor tasks, versus those that relate more to attention in the middle of the gradient, versus those that relate to social cognition and emotion. So you see here this pattern from blue to red. And it's important to note that regions that are far apart may still belong to the same rule. So here the red regions in the default mode network, they're very dispersed across the brain, but still they have something about them that is similar. And they're also co-activated in task. And you also see that to some extent, they also have similar microstructural features, roughly. But there's more devil in the details, but this is more an overview to get you curious. And this pattern also relates to anatomical hierarchy as defined by T1, T2 weighted ratio in the brain that is a proxy for myelination and a couple of other things. And you see that it's also similar in macaques. Then again, it also relates to an evolutionary hierarchy or at least differentiation, because it's important in evolutionary study not to see humans as the best version of a brain that could come out of it, but just one variation of how a brain could turn out to be dependent on the environment that a certain living being has to function in, right? So a human would be kind of unhappy if they had a penguin life, right? But if you were a penguin, you wouldn't be standing here where I'm speaking, because that would make you very unhappy, and you're not really made for this. But it's important to note that you see always also these same principles coming up where you have sensory areas and some association areas in between. So it seems that these are in some way basic building blocks, but then they're mixed and matched depending on, well, the creature that we're looking at. So what does it mean? So for this, we have to change systems and skills and also ideas. So I already spoke a bit about continental drift. I spoke about penguins. And I spoke about structure and function, and this is kind of what I try to do and what gradients, or how I apply gradients to kind of build bridges between different ideas and different disciplines, and also between the microscope and ultimately the MRI scanner and behavior. I'm not sure what comes here. Oh yeah, sorry. So there's many people that like this and that apply these different techniques. So over the past, well, since 1988, I was just born. there were like already a couple of techniques that really differentiate different brain organization or approaches or to study the brain from the level of the brain towards the level of the synapse and to the level of time, so millisecond toward months. So it's important that if we're dealing with brains and we want to understand how they work, we also have to deal with space and time and make these connections. And as you'll see, there's some overlap between these methods, but it's also, you know, to build bridges, it's not a complete like similarity between, for example, EEG and electron microscopy. So, then the basic workflow of a gradient approach would be that you have a functional connectivity matrix, or in my case, a genetic correlation matrix. It can be any matrix, really, which we will also highlight later. And then you do an affinity computation, such as cosine similarity, and you create an affinity matrix showing how similar the rows are. And you order them. So you want to understand which rows are most similar to each other versus which rows are most distant to each other. And for this, you get, like, for example, two gradients are displayed here in the first component, gradient 1 and gradient 2, and you project these on the surface. And then either you study the spatial patterns, but you can also use it as a way to understand individual variation along these low-dimensional principles. But there's a lot of caveats that come with it if you start thinking about how also to align across individuals, because it's not so easy to define what is most similar if you think about these low-dimensional spaces. At least in the end, you get a sorted connectivity matrix, and with this, you're happy. And what is important here, and I will also highlight it later, there's a lot of analytical choices you can make here. So what you do, try to break what you find, is maybe the advice that I also learned from Amin, because you want to know the boundaries of your result. So if it's only a significant finding, if you take a certain set of features in a certain context on a certain morning and you cannot restart your laptop even because you're afraid you won't find it again, reconsider what you're doing. So what I usually do is also, because there is no ground truth in these methods to some extent, I will do a PCA, but also a nonlinear embedding, and see to what extent these gradients remain similar no matter what I do. So I also often like to change different atlases based on Schaefer, but also based on Matt Glasser's atlas, to just see like what kind of features redefine my pattern. So this is, for example, something that I did with this genetic gradient project. So we're now going to hands-on part 1, which is the next challenge but we'll do it. I mean, it's... okay.
Is it down? Yeah. Okay.
Will you speak, or will I...
we
can do, like, I do one sentence, you do... Okay, okay. So Amin is is helping me, and shall we go here on the screen to the hands-on, or?
Okay, so I will now should change the screens. Okay, so first of all, before we change the screen...
Take your
if you
phone.
want to walk along with the, sorry, maybe I should stand up. If you want to walk along with the tutorial, scan the QR code. Can you also then switch it again back to the other? No, sorry. Just follow along. But if you don't, it's there.
You can also passively sit and see me make mistakes. So
yes. But if you want to follow along, just do it. And then there is a setup cell. It's going to take a while to run. So. And then it will ask you to restart, and then restart and run again. So I've already done this for the interest of time. When you open this, you should create a copy of the notebook for yourself and then start it. And can we switch back to the other screen?
Yeah, the copy part is important, right?
Sorry?
It's important to copy it.
You would have to copy if you want to make changes, or if you want to keep your changes. But if you don't also make a copy, it's going to work.
Okay, thanks.
Yeah. Okay, so first there are 2 setup cells. Like the first setup cell just installs the things that are necessary. And then the second setup cell just defines some functions. If you're interested, just look inside them, see what's there. We are going to use those functions in this hands-on. And now in this hands-on, it's going to be 3 parts. Part 1 is going to be about building a gradient. So we just explained that we are going to build this gradient first for one subject and then for a group. And then in parts 2 and 3, in part 2 we are going to contextualize this. And maybe we should talk about part 2 and 3 when
we just do it when we do it. So
okay,
we'll see what we manage in the time. But otherwise, kudos, he made very clear tutorials, so you can always revisit them during the Taliarch lecture, no, or whenever you find time. So yeah, maybe we get it started. So I say what I do and you do it, or how do we do it? Like the steps, or you want to talk it through? Maybe
I can talk it through, and if
Yeah,
you
yeah.
have any points to add, then you can add them as well.
Okay, yeah, no, that's the best
Yeah.
distribution of, yeah.
So first of all, like shout out to Brainspace and to Brainspace tutorials, because the gradient part is based on their tutorials a lot. So check out their tutorials as well if you want. We are on the setup cell. So in the first part, so we are going to create a gradient. We are going to create gradients of functional connectivity. In part 1, we are going to create this for a example subject. And part 1 is to just build the functional connectivity. To build a functional connectivity, we need a bold time series. I'm going to skip or walk quickly through this because a lot of you may be familiar with this. So we are going to load some example bold time series from Brainspace, which is included there. So we need the time series, and we need also confounds, because we need, as you know, like bold time series needs to be cleaned out of confounds, like such as motion, etc. So we also load them, and then we now have time series. These are time series. By the way, like what we do in this tutorial is all at the level of parcels. You can basically do these also at the level of vertices and cortical surface, but the reason that we did this on the level of parcels of one of them was that it's just more manageable to do it in this tutorial, also like in general in practice, because it will require a lot of calculations to do this on vertices. Yeah. Yeah. So we have the time series, we have the confounds, and now we use, I guess, nylearn signal function, signal will clean, to clean the time series out of the confounds, which is just basically going to regress them out. And the next part is just you just label them, like basically. So the way that it works. Sorry. So this time series is actually at the level of, I think, parcels of vertices. We need to first parcellate it. And this bit does this parcellation part. And eventually we have like this parcellated time series which we can work on. And then next part, maybe more interesting part, is that so now we have this parcellated time series, and now we are going to create a functional connectivity matrix. And there are like basically to create a functional connectivity, you just correlate, and I mean you could basically also use NumPy correlation function. But you can also use like this nylearn connectome function, which offers more functionality. And then now we have this correlation matrix, which is the functional connectivity matrix for this particular subject, using a very coarse-grained parcellation, which is Dijkstra's. And then we now are going to plot it, or it was already plotted, but I'm going to run it again to see how it works. So we're just plotting this matrix. So this is the functional connectivity matrix. Is also very big. So then this is what we are going to work on in the next step to create a gradient to make sense of this, because this is a matrix that we cannot really make a lot of sense from. So one reason that we make gradients is this, to make sense of these matrices. Okay. So then, now we have this functional connectivity matrix, and now we are going to create a gradient for this particular subject that we just had the data for. And so for creating gradients, we are using Brainspace. It comes with this class, gradient maps. And we are going to go with the defaults and create 2 components. But check out the documentations. There are a lot of options, as Sofie mentioned. It's a good idea to try out different options to see how it works. So then the way it works is that you create this and then fit the FC, functional connectivity matrix, to this gradient maps object. And then eventually you would get like these gradients, and then you can visualize them. And the way that when you the thing that you see here, these are the 2 main axes. that would describe the variance in this function connectivity matrix. And here, as Sofie mentioned already, like here these maps show which regions are more similar to each other based on their functional connectivity in different axes or different components. And here you're seeing like two components, two principal components of the FCE from this subject. And just one note about like plotting is that so our data was in parcellated space, so we need to map it back to the vertex space, and then there are a dozen ways to do that. One way is just to use this function map to labels from brain space, but you can also make your own function. It's basically just to know which vertex belongs to which parcel, and also what is the value of each parcel. Then you can just map back two vertices. Just very simple. Yeah. So this was one for one subject. But then we want to. This is like one gradient or one subject. But to be more precise or more, just more robust, we need to go to the group level. And in the next part of the tutorial, we are going to go to this group level and do the same step, but using not the FC of a single subject, but an FC that is from the data of many subjects. I don't know how many though. And again, brain space has this sample data from the sample group average function connectivity that is come in the toolbox. And then here we are going to load this into this connectivity matrix by calling like this function. And we are here using, like based on the tutorial, we're going to use Schaeffer 400 atlas, but you can also choose different atlases. Just check out the documentations. And we are also going to load the parcellation map, again for the purpose of plotting later, again for the mapping that I just mentioned. And this is also, we are going to plot this on a different surface, that doesn't matter. And again, using the same object, using the same class, gradient maps, we're going to now create gradients. Now, this time we are going to create 10 components. But we are going to just plot the two. Yeah, again, check out the options. See, like, you can vary them. And when we create gradients and plot them again with the same approach, we can see these two principal gradients, which are actually like what was described first by Daniel Margulis, who is also here, in 2016, but that was done at the level of vertices. But when you look at it at the level of parcels, it would be look more or less similar. Yeah, right. Yeah, so that's it. Like, I mean, we went from a function connectivity matrix for a subject or a group and tried to summarize it in a gradient map. And again, this gradient map, what it shows is that similar colors have similar connectivity profiles or are connected to similar regions in each of these like different axes. And what else?
I think we have a question. Or are you just standing there?
No.
Okay.
I mean, it's better to view from here, but still, I do have a question. It concerns the input to everything. I mean, I see you calculate the functional connectivity matrices in this example yourself, but say I already have functional connectivity matrices. Mm-hmm. Can I just plug them in? Is there a specific format that this needs to be in? Is there anything that I need to look out for in the data before I bring it to plotting gradients? Can you say a little bit more about like what the data need to be able to work with this?
Shall I go? Yeah. You just need I think it's pretty basic. So either in MATLAB or Python, you just need a matrix. And you also need to know the labeling of that matrix, right? So either you use like Schaeffer, which is already in the toolbox, or otherwise you have to have your labeling either in FS average 5 or Contra 69. But yeah, you can just plug in any matrix. You can also do a gradient of any matrix and then not plot it on the brain, right? It's just a dimension reduction technique ultimately that is put into a toolbox. So it's not so complex, and I think this is also a good jump to the next part, because that is also the tricky element of it, is that making these gradients is like, my kids could do it, right? But that doesn't mean it's scientific to create a pattern. So having made it to this stage, and probably you're all a bit like underwhelmed, the hard part comes because, like, what do these things mean beyond the obvious? Because right now, like, a lot of people say, differentiation between sensory and transmodal, but yeah, what else is new? Like, this was known already. So to kind of make further understanding and to move science forward, it is important to think about these gradients and use different measures or different skills or different techniques or different contexts to interpret what you're seeing, and also to cross-validate what you're finding with one method with another method, to see how do you find harmony or synergy in different ways to look at the problem. At least this is what I usually do, because I think a gradient alone doesn't give you, it gives you a nice bachelor thesis if you need to supervise somebody, so it's a great way to keep people entertained and like teach them the first steps of computational neuroanatomy. But if you want to do research, you need a bit more. And that's why we're now talking about the bit more part. And we go to the, we're already back to the slides. So decoding 101, and this is what I said, like, it's called decoding, and this is also still not mechanistic really, like it's in the end often just a spatial correlation of one pattern and another pattern. But it can also relate to like individual difference modeling using linear models or fancier models than linear models. And it also could link to linking genetic expression patterns to your brain patterns, or histological patterns to your brain patterns, and or meta-analytical patterns to your brain patterns. So as you see, this is all still far, far away from a mechanistic understanding, because again, it is only correlations. But that doesn't mean it's not relevant. And sometimes it really helps to also use these correlations to get a motivation to really dive into the details, because you want to know how this correlation came about. And for this, you can, for example, use BrainStat toolbox, but there's a lot of like NIMARA, there's a lot of toolboxes out there that are made to look at meta-analytical maps from different resources. And I will also discuss some other toolboxes with genetic expression to some extent that help you do this decoding, because also here it's important to note that there's so many ways to process data and to look at data, and all of these ways give slightly different results, making it also very hard to know the ground truth, and also it's important to know the stability. So, yeah. So what is genetic expression? So genetic expression is based off microarray data, and based on this, the encoding of the genes is sampled through mRNA and proteins, and then you need to do a couple of steps to know from the raw data if it's like meaningful or also noise, similar to what we know maybe from fMRI and structural imaging, or all data processing techniques that we use. And this is also the tricky part, that just because it's available, you still need to know what you're doing to get use of this new modality and this new idea and integrate it in your findings. And thank God, there have been amazing people. For example, Arnett Kivichuye. Okay, I cannot pronounce this name, but they wrote a very nice neuroimage article in 2019 describing the different steps and setups that are important to consider, including also data filtering, probe selection, sample assignment, so to understand like how a certain probe relates to a certain brain region, data normalization, and also gene filtering to look, for example, at the consistency of the expressed gene, and also then potentially a region times gene genetic expression map comes out of there, and then you can also account for other spatial effects. So there's a lot of steps, and this was the first paper highlighting this, and then also a next paper came by Ross Marcello, where he also made a toolbox, Abajan. So this toolbox gives a lot of answers, but what is interesting, if you see that if you have these analytical approaches where you can look at either correlated gene expression, but also gene by gene relationships, but also regional gene expression versus T1, T2 weighted maps, or a PC1 of gene expression, or a functional gradient, there's all kinds of different things you can do once you have these data, right? And you can relate everything with everything once you know where to find the correlation function. However, depending on how you filter and how you set up your analysis pipeline to get to clean your genes and know what is noise and what is true, you also see a high variability in these different types of analysis. So in B, you see actually the correlation values vary a lot with the approximately 7,900 different options you have in this analysis pipeline. So it's really important to consider state-of-the-art and recommended design steps to do this kind of genetic or transcriptomic neuroimaging to be sure that you're doing something meaningful. So like I can really recommend this toolbox because they really nicely kind of set this up so that even like people that are new to the field kind of at least get to understand the basics and hopefully don't make the basics wrong and then draw wrong conclusions of it. So it's a super nice eLife paper, and also the toolbox is very helpful. And then you also see that depending on what you do, you see also slightly different results. So this makes it also tricky sometimes to interpret whatever is being said, because it's also quite volatile to certain decisions. Yeah, also Conrad did great work also in using the Allen Human Brain Atlas and linking this also to spatial smoothing to get like at a higher sampling. And there's a lot of people really thinking about how to really use this resource, which was like ultimately 6 donors that were used. Yeah, of course, this is a huge sacrifice, and we want to value this in a good way and handle it in the right way. And recently also Richard Deere, he cleaned the genes in a certain way, looking at 3 principal components and not 1. And then he found 3 different patterns of genetic co-expression. Also, kind of gradients or patterns, as you see here, on the one hand differentiating more sensory from paralimbic areas, as you see in C1. But then you also see like more visual to frontal pattern changes, but also another pattern going more from sensory visual to what it is, default mode network regions. And these 3 patterns that came out and after a certain stability check also had a different biological meaning if you consider layer enrichment, but also if you link this to other Go enrichment patterns. So by doing different processing techniques and thinking really carefully about the meaning and to get the most pure or most true data, you can also dive a bit deeper into the different dimensions or gradients that relate to the co-organization of different features, such as genetic expression. So another way to enrich your data or to consider how structure or functional data relates to other features is to consider receptor distributions. So Justine Hansen also made this available through neuromaps. And she had the amazing collaborative idea to collect all kinds of PET distribution maps, for example, from serotonin, dopamine, norepinephrine, histamine, acetylcholine, cannabinoid, opioid, glutamate, and GABA. And you see that all these patterns vary across the brain, and this is also an important interface between anatomy and function, and so it's important, or it can be valuable, to link this into your patterns and also to understand how these features co-vary across the brain, and also like how this organization may help us derive certain patterns you see in patients or certain developmental trajectories. Important note is that you have to be aware of space, because you do a lot of transformations either from an MNI map to FS average, FSR, LR, or CFAT space, and then you have to compare this to spatial null models to be sure that you don't find a random spatial correlation, but that this is actually specific to your map. So there's a couple of techniques now going on, and also the field is developed. There's also an eigenmode way of doing accounting for spatial null models, but what we use a lot is, this, what is it called now?
Spin test?
The spin test, sorry, thank you. I knew this word, I've used it myself. So you spin the brain, so to say, around its axis, and with this you have like 1,000 randomly spinned patterns, and you see whatever spatial correlation you have is more significant than, like, depending on if you do 1,000 tests, 950 of these random patterns. So with this, you kind of also account for certain statistical challenges. And we will now go to hands-on part 2. I don't know how much time we have. 5 minutes. Would that fit?
Yeah, we kind of skipped the third part, maybe.
Or your talk about, do you prefer theory or practice? Maybe. we can.
Maybe we can do the practice, right? Or
Yeah, we do the practice quickly.
okay.
Yeah.
Sure. Yeah. So can you switch to the other? Yeah. Okay. Now you have to look this way. Remember the map that we just made? I'm going to just also rush through this. This map. We want to see which genes and which pet maps it's associated with. This is the simplest thing to do. Here, I mean, you can do a lot of things with this sort of data. In a way contextualize it. Again, you can do this at the level of vertices, like both with the gene expression data and also with the pet maps. But here we are going to do this at the level of parcels. A word about like gene expression data. Sofie already mentioned this. Like it comes from this Allen Human Brain Atlas that you can process yourself, but you can also use tools such as Abagen or similar tools to process. And we have already done this because it also takes some time to download and to process, and made this available through this link here, or actually it is like from a previous project of ours. And also like this is a sample code of how you can get this data, but it's also more complicated a little bit. There are again options in Abagen in how to process this data. So for example, you have to select an atlas or other options. You can just check it out, check out their tutorials. They explain all these steps, all the decisions, but then we went only with the defaults. Eventually what you get out of this is that you have like 15,000 genes and their expressions in like the parcels, but here we have only the left hemisphere because the right hemisphere data is very sparse. And then we can basically bring our, wait, I didn't load this one. Oh, come on. Okay, sorry. Okay, maybe I don't run them because of time. So we load these like genes and then also like again have our gradient in the same space. So both of them are parcellated, both of them are aligned. Now we can associate them, and there are different ways to associate them. One way is to run a PLS, select the top 500 or top 1,000 or whatever genes that are associated with them. So we can then see like which genes are positively associated, which genes are negatively associated. And we did that, for example, for this one. So these are the genes that are positively associated with our map. This is the average expression of them. And we have like, how many? 200 something genes positively associated out of the 500. And we also have genes negatively associated with this map. And now we have two sets of genes, and we can do all sorts of things with this. One thing that we can do is just use these gene enrichment analysis, for example, a developmental enrichment. There is this online tool, check this out. But I have here wrote a function to like parse this, like go send this request to this website and then get the results back about the developmental enrichment. You can check this out. And eventually what you can get is that like which developmental stage and which structure these genes, these 200 something genes that are positively or negatively associated were enriched in which stages, and also like a sum of all the p-values, but never mind the details. And this is also negatively associated genes. And then it's like make inferences about what's different about them. And then moving on to the pet maps very quickly. So the pet maps, as Sofie mentioned, like they are stored in NeuroMaps toolbox, which is a great toolbox. And it comes with datasets and also tools to work with these data. And the atlases that we got from these pet maps are in MNI space, so we had to parcellate them again into the same parcellation. And again, if you want to work on the surface level, you have to transform them from MNI to the surfaces, the different spaces. And then again, this is, I have already, like, you can get the data that I have pre-parcellated here from this link. And then you can load this, and you can, then we had merged them because some tracers, you have multiple maps of them, so you can merge them, take an average. And then you can also plot them. So these are different, these are the names of the tracers, not the receptors themselves, but you can look them up which of them relates to which receptor. And here are like the four example ones. And then we can do association between the gradient map and these PET maps, which are like 20-something PET maps. And in association, as Sofie mentioned, you have to be careful of spatial autocorrelation. So to account for that, you have to do something like a spin test or variogram test, or there are different methods that exist out there. And here we are, again did this spin test at the level of parcels using Enigma toolbox, which is again a great tool. Check that out as well. And this is like a spin test parcel function which you can do. This was in cell 2, the hidden cell, of how it works. The way that it works, it creates permutations and then that run the test and then compares to a null distribution. Anyway, you eventually see like which of these genes are significantly associated or not. This is a way of plotting this. Like these are, sorry, I said genes, receptors. So these are, we have some receptors at the top, negatively associated, and some at the bottom, positively associated. And with this, we can make more sense of our map. What does this mean in terms of biology and gene and PET maps? Yeah.
Yeah, so I think then we also come to a close. I think the most mechanistic part was actually a toolbox that Amin also developed. And if you check my Twitter, like we recently also did a QBAMN toolbox to translate from GPU to CPU processing for excitation inhibition modeling based on the structural connectome. Which goes deeper maybe into potential more mechanistic and computational models, understanding these patterns of brain organization and how structure relates to function. There's also a QR code and a Kaggle. So I will put this also on Twitter, or we make this all available for everybody. So if you want to get a start, at least to start thinking and trying about bridging skills, using gradients, and thinking about why a certain area is where it is from different angles. Check out the code, and yeah, thanks Amin, and thanks all the technicians and Matt to save the day by plugging. Yeah, enjoy the rest of the session, and thank you.